Sorry, I could.
That's great. So, hola, Carmo.
It's all good?
It's all good. Do you hear me? Hola.
Yes, I can hear you, and you can hear me well?
Yeah, we hear you. Absolutamente fantastico.
Great. I'm
fantastico persona. Carmo is an outstanding person. As a matter of fact, Carmo and I are almost soulmates. We also born a few days apart on the same year. And throughout the network, the European network that we are involved with, every time that we are meeting together, it stands out that we are coming to the same conclusion. So it was really a great pleasure for me that a few years ago, Carmo came to visit my lab, and it turned out that we can collaborate, and it came out in a wonderful molecular cell paper that I'm going to talk about it today. So Carmo got a very unique disease. She was born in Portugal. During the dictatorship, it was extremely hard to get out of Portugal. Nevertheless, she was managed to go to EMBL to do her postdoc. She came to a lab that's working on chemistry. She was working on 2'-methyl oligos. And then she met, during lunchtime, Angus Lamont, that was a young PI, and he was working on small nuclear RNA, the stuff that Adrian talked about it, and they decided to collaborate. And within 2 years, there was a barrage of papers in which Carmo used anti-space oligo in order to probe the small nuclear RNA, and they found structures and more structures about the recycling of snRNA. Maybe she's going to talk about it today, or maybe not. Carmo returned back to Portugal, became the #1 scientist in Portugal. She was the head of her institute. She is a top scientist in cell biology, microscopy, immunofluorescence microscopy all around the world. She did also a lot of bioinformatic analysis, and there is no one that is more appropriate after Reinhard Newman to present the cell biology aspect of the splicing machinery. And Carmo, the podium is yours. Thanks a lot for being with us.
Thank you so much for this extremely nice and kind introduction, Gil. I'm so sorry I am not there, but the lesson that I have learned is that these days we cannot trust air traffic control, and I'm now trapped in Munich airport, and the only way I could present this talk in a timely manner is by Zoom. So my deepest apologies, and I'm sure we'll meet up in the near future. So what I will talk with you today is about the spatial organization of splicing in the cell nucleus, and I'm proposing to make a journey from test tubes to live cells. But before starting, I would like to remind you that our view of gene expression has changed dramatically over the years. And not so long ago, in the 70s, when there was the bloom of the birth of molecular biology, and many of the early molecular biologists were studying mainly bacteria and viruses, the idea back then was that the lessons that were learned, the knowledge that was learned from these organisms, from bacteria and from viruses, would probably be conserved and could directly be transferable to higher plants and animals. And according to this view of making predictions about what you don't know based on what you already know, the prediction was that mammalian cells would have up to 2 million genes. And now we can smile at these and realize how predictions based on our current knowledge can be totally wrong. Also, the discovery of interruptions in eukaryotic genes, basically the discovery of splicing in 1977, came as a complete shock because this was not present in bacteria, and therefore, again, the assumption that gene expression is very well conserved began to disentangle, and now we know that the control of gene expression has evolved in very complex and diverse ways. So, what we know about also what our perception of splicing and the complexity of splicing has changed over the years, very much based on the technologies that have been available to address these questions. And you can see that if you would ask the question how many human genes are alternatively spliced back in 2002, the answer would be approximately 60%. More recently, after 2008, we are aware that practically all human genes are alternatively spliced. But what do we know about the origin of splicing? And when scientists go back and try to find the first evidence of anything related to splicing, some sort of ancestor to splicing, what comes up are particular types of non-coding RNAs in bacteria and in mitochondria that contain self-splicing introns with characteristics of mobile elements. So the current view, but obviously this is an hypothesis, the current view in the field is that introns probably arose from the invasion of an early eukaryotic ancestor by hostile genetic elements that had encrypted within them the means for their removal. But once introns entered protein coding genes, they never left. And we see that in the most simple yeast cells, we find a small percentage of genes containing introns, and throughout evolution, as organisms become more complex, we see a progressive increase in the fraction of genes containing introns and the number of introns present per gene. Now, most of what we know concerning the mechanistic reactions that underlie splicing have been based on in vitro splicing assays. And basically, how you do these assays, you do an in vitro transcription of an RNA, and then you mix this RNA with extracts prepared from cell nuclei. And what you initially, you start with a pre-messenger RNA, an RNA molecule that contains introns, and in the test tube, splicing occurs, and the intron is removed, and you form a mature messenger RNA. And because in these assays, you start with a previously transcribed RNA, a conclusion that you take from these experiments is that splicing does not require transcription in order to occur. But while a lot of biochemical labs were using extensively these splicing assays, another lab that was more focused on using electron microscopy to study genes and the organization of gene, and this is the lab of Oshine, where Ann Bayer did these beautiful spreads of eukaryotic genes and look at them by electron microscopy, and you can see this axis here corresponds to the DNA of the gene, and these lines here, this is the actual picture in the microscope, and this is the interpretation of what you see in the EM image. And these lines here correspond to the RNA that is in the process of being synthesized. And this is the direction of the synthesis. So these were the oldest RNAs that have been synthesized, and these are the youngest, the latest RNAs to be synthesized. And when Anbeyer and Olsheim looked carefully to the length of these transcripts, they realized that some of these transcripts contained lariats. You could see the shape of a lariat. And then when the transcript moved to the next step, it was shortened and the lariat was no longer observed. So the fourth and the fifth position means an early RNA and a later RNA. So they interpreted these as this lariat corresponds to the splicing reaction, and now this subsequent RNA no longer contains the intron, so it has been spliced. And this was really the first convincing evidence that in cells, intron exclusion can occur as the pre-messenger RNA is being transcribed. And also because you can measure this distance between the intron containing and the spliced RNA, you can measure how many nucleotides are between the two, and we know what is the rate of transcription of Pol2. So taking into consideration the how long it takes to transcribe, this electron microscopy study suggested that intron excision could occur within 2.5–3 minutes after transcription. And this was in clear contrast with much longer times required to go from pre-messenger RNA to mature messenger RNA in the in vitro assays. But timing being slower, at least tenfold slower in vitro, was not the only difference. Because despite many attempts, these in vitro assays are not good in recapitulating alternative splicing, and also they tend to be restricted to only short introns. And yet we know that in mammalian cells, a large fraction of introns are very large. In fact, over evolution, introns tended to expand, and humans are kind of champions of having the longest known introns among the sequence vertebrate genomes. So something in the nucleus is clearly important for the splicing as we know it and happens. So the hypothesis is that spliceosome components in the nucleus are probably organized in a way that favors splicing. And the big question is, how can we address this model? How can we tackle the three-dimensional organization of the spliceosome components? So I will briefly go through historical milestones that many labs, including our own, followed to address this question of how are spliceosomes organized in the nucleus. And so, as Adrian already told you, the spliceosome is a multi-complex machine, and major components are the so-called SNRPs, that contain small RNAs and a number of associated proteins. In early studies, we're using antibodies against the different proteins of the SNRPs to do immunofluorescence and localize them. But as Gil already mentioned, I was extremely lucky to be at the MBL in Heidelberg exactly at the time when Angus Lamont was working with a chemist called Brian Sprott, and they were developing oligonucleotide probes that were complementary to the RNA component of the SNRPs. And because I had a microscopy background, I decided to test those oligo probes, adapt them to a mix between the in situ hybridization and immunofluorescence. And in fact, the results were quite surprising because we found these bright foci in the nucleus concentrated of signal against the RNA of the SNRPs. So this immediately attracted a lot of attention because SNRPs are the main components of the spliceosome. So what would be the relation between this new compartment and the splicing reaction? It turns out that this was not a new compartment at all. It had been, this structure had been described back in 1903. These are original drawings by the Spanish histologist Carmo-Fonseca. And when he was looking at neurons, he was developing different staining methods. And he had a particular staining method that highlighted the nucleus and these dots that he already described as the dot. And basically, subsequent studies showed that the dots that we identified enriched in the spliceosome snRNA was exactly the same structure that Ramon and Cajal had identified in neurons. And that's why these bodies are now called Cajal bodies. Many studies, as Gil mentioned, many studies were subsequently done to understand what's going on in Cajal bodies. And the current view is that these are sites where SNRPs are pre-assembled, are modified and pre-assembled before they are ready to enter the spliceosome. So there is no direct link between these structures and the splicing reaction. But this is not the only place where spliceosome components are visualized in the nucleus. In addition to the Cajal bodies, to these bright dots, spliceosome components are not homogeneously distributed throughout the entire nucleus, and they tend to be enriched or accumulated in these irregular regions that have been called nucleus speckles or splicing domains, because they contain basically all of the components of the splicing machinery, not only all SNRPs, but they all contain all the other factors that are required for a spliceosome to assemble and to catalyze the removal of introns. So, but this still does not tell us where splicing exactly is taking place. So another methodological development that was important for the field was the development of fluorescence in situ hybridization. And my own lab, when I returned to Lisbon, was among many labs that were adapting fish to image genes in their transcripts. And this is one of our early papers where we could show, in this case, we were looking at the organization and the transcription of the beta globin gene. And these are images where you see the locus, you see where the gene is, using a DNA probe for the non-coding, so for the regulatory sequences associated with beta globin, and then a probe that spans the coding region of the beta globin gene. And so what we see here is the RNA. And in this overlapped image, you can see that we can detect the nascent RNA coming right from the gene. In this other cell, this cell is not transcribing, so the gene is there, but there is no RNA because it's not being transcribed. And what fish allow us to do is to design probes that are complementary to different exons. So a probe that hybridizes only to exon 1 will not give any signal, but because the probe has a sequence that is complementary to exon 1 and the rest of the sequence is complementary to exon 2, when the exon 1 and exon 2 are already spliced, then the probe can hybridize and give a signal. And so by designing so-called splice junction probes, we, as many others, could see that spliced RNAs are already detected at the site of transcription. So splicing is occurring co-transcriptionally, again, in accordance with the early electron microscopy data from Bayer and O'Shine. But now that we can do this in a fluorescence microscope, we can establish what are the relations between the genes, the site of transcription, and the splicing domains, the nucleus speckles. And work from many labs showed that indeed there is an intimate link, an intimate spatial proximity between active genes, so genes that are producing RNA, and these RNAs are being spliced, and these RNAs are being spliced in very close contact or embedded at the periphery of the nucleus speckles. Probably the nucleus speckles are providing this scaffold of enriched spliceosome components to allow splicing to be so efficient. In this intimate connection between genes and speckles is lost when genes are not active, either when we look at genes that are not transcribing in a particular cell type, or in a cell type treated with transcription inhibitors. This association is lost, reinforcing the view that indeed the speckles are providing the help of spliceosome assembly to the newly transcribed RNAs. But all of this so far is done on fixed cells. Can we move on and look at splicing in live cells? And this became possible later on at the end of the 90s. And this type of technologies were pioneered in the lab of Rob Singer and David Spector, and many of the postdocs who were in Rob Singer lab then became independent group leaders like Edward Bertrand, Aaron Shaftal, that you will hear in the discourse. And all of these scientists became experts in living cell analysis of RNA biology, gene expression. So basically the method consists of taking advantages of sequences from bacteriophages. In this case, I'm representing here the RNA sequence of the MS2 bacteriophage, which forms a stem loop that binds very tightly to a protein that in the phage is part of the coat of the phage. But what we take advantage, what we engineer, is we clone the sequence that is transcribed into these RNA stem loops into our genes of interest. And then we express in our cells the protein that binds to the stem loops, and this protein is fused to a fluorescent protein, like a green or a red fluorescent protein. In the early days, you would clone these sequences in reporter genes, and that's what we have done when we did this work. Nowadays, you can obviously use CRISPR and introduce these sequences directly in your gene of interest, in your endogenous gene of interest, in your preferred cell type. So, because we were interested in visualizing splicing, our idea was to introduce multiple copies of these stem loops. The reason why you need multiple copies is that you need a sufficiently bright signal to be seen in the microscope. And so we introduced 24 copies of stem loops. We express this reporter gene in human cell, and we look live where is the signal. And basically what we see under normal conditions is we just see one dot, one fluorescent spot in the nucleus that corresponds to the nascent transcript that is being produced at the site of transcription. Something that is very important to bear in mind is that when you introduce these multiple copies of MS2 or any other reporter, you must make sure that your reporter, your tag, is not interfering with the process that you want to analyze. And this was indeed the case. We realized that if we introduce the MS2 stem loops relatively close to splice sites, then we were inhibiting splicing. A lot of the tagged RNAs were not correctly spliced. But when we increase the distance from the splice sites, then the vast majority of our tagged RNA was correctly spliced. And another way to control for the correct gene expression is to clone a small sequence that when the mRNA is transcribed into protein, this introduces a peroxisomal targeting site. So the protein will be targeted to peroxisomes, and this is fused to another fluorescent protein with another color. So if we see cells full of magenta peroxisomes, this means that our reporter with the tag in the intron was correctly expressed in a protein was correctly made. So what we see when we follow in these live cells the lifetime of an intron, we only see a spot in the nucleus, as I told you, but we see fluctuations in the intensity of this spot. Sometimes there is nothing, then a few seconds later, a spot is visible, then the spot remains for some time, then it starts to get dimmer and dimmer until it disappears, and then signal comes back, and we see again spot of light, it becomes dimmer, and then another round of light comes again. So basically what we are seeing are fluorescent fluctuations in fluorescence intensity. And the way we interpret these fluctuations is that every time we see a signal, this means that the polymerase has transcribed the intron contains the MS2, the MS2 bound the GFP fused protein, and so we see fluorescence. As the intron is removed and it either moves away or is degraded, then the fluorescence disappears. And because we don't see introns floating around in the nucleus, except when we inhibit splicing, we presume that intron degradation occurs close to the site of transcription, and therefore the fluorescence disappears without seeing introns floating around. Once we have these measurements of intron lifetime, we can now compare introns with different characteristics by engineering reporters with different sequences. And our first experiment was to change the sequences of the splice sites, because from in vitro-based assays, it was well established that some sequences are closer to the consensus and are called the strong splice sites and are expected to lead to more efficient splicing, where the sequences that are less conserved are called weak splice sites. They tend to be more common in alternatively spliced exons. And the idea would that these weak splice sites would take longer to be spliced because they are not so efficient. And indeed, we were extremely excited to see that when we compared the intron lifetime that is measured here, and here you see the statistics, when we measure how long the spot of light remains at the site of transcription, when we are looking at reporters with the weak polypyrimidine track or a strong polypyrimidine track, the weaker splice site, the weaker splice sequences were taking longer to be transcribed compared to the strongest one. So our assay was capable of resolving differences in timing of splicing, but still everything is within seconds. Everything occurs within seconds. The making and the degradation of an intron occurs on average in 30-40 seconds. Then we thought, what about if we change the length of the intron downstream of the MS2 binding site? So as soon as the MS2 binding site is transcribed, then the GFP can bind and we see a spot. But the polymerase must continue to transcribe the downstream sequence of the intron until it reaches the 3' splice site, and only now the spliceosome can assemble and the intron can be removed. So we thought if we introduce extra nucleotides in the intron, then the polymerase should take longer to transcribe these extra nucleotides before the signal disappears by splicing. And indeed, this was the case. By extending the downstream length of the intron, we could prolong the lifetime of the intron. And when we make some calculations, we have introduced one extra KB in the intron. And according to the established estimated transcription rate of POL2, 1 kb will require 20 to 15 seconds to be transcribed. And when we see the difference between the lifetime of the short intron and the long intron, the difference is exactly within this range of about 20 seconds. So if the difference is the time it takes to transcribe, this means that splicing must occur with just a few seconds that we are not being able to resolve in these experiments. And so basically this was our most surprising conclusion when we first published these results back in 2013. Splicing can occur within a few seconds after transcription. And I want to highlight to you that exactly in the same year, there was this unifying review published by one of the early founders of RNA molecular biology, James Darnell. And according to James Darnell, still splicing requires several minutes for completion, because this was the current perception in the field. So coming back to my original idea of this talk of going from test tube to live cells, I hope I exemplified to you how important it is to approach a biological problem using different methods. In vitro splicing assays are absolutely critical and fantastic tools to uncover splicing reactions and the mechanistic details, but they are not telling us the whole story. In splicing in vivo can be in the order of magnitude of 100 fold faster. And now obviously this raises a lot of questions that many of us in the splicing field are trying to pursue. And I would like to acknowledge the people in my lab that were involved in this live cell imaging work. And a special thank you to Tom Kirkhausen from the Harvard Medical School. This work was initiated when I was doing a sabbatic in Tom's lab. And without that, it was impossible to have done this work. And right now, I would like to leave you with a challenge, which is, I'm sure that each of you now are thinking about what still are the big questions that I would like to know about splicing. And I hope I can help discuss these with you. So thank you very much for your attention.
Thank you, Carmo, so much for arranging such a lovely talk within a very short period of time. Questions? Sure that so can you a little bit elaborate about the Cahal body with respect to the function in terms of what the snRNP are doing in the Cahal body since you discovered them? What was known since then?
Yes, so what was mostly discovered was that spliceosomal snRNPs, they have a very complex cycle. They are by themselves small machines made of one small RNA and a large number of proteins. Many of these small RNAs are chemically modified, and depending on the chemical modifications, the proteins that associate can or not associate, and this will trigger their ability to participate in subsequent reactions, namely in active spliceosome assembly and catalysis. In these steps of SNRP assembly are occurring in the Cajal body, and a very important component, a factor, a protein factor that is related to this SNRP assembly is the SMN protein that I'm sure Adrian already referred. At some point in the field, there was a confusion between Cajal bodies and gems or Gemini, because some localizations of the SMN protein look like foci next to Cajal bodies. So this created again a lot of confusion and a lot of need to understand what is the role of these different compartments. But basically, the related function is pre-assembly of SNRPs, of spliceosomal SNRPs. Another question, another point that I think is normally asked is whether you need a body to do this pre-assemble, whether the compartment is a consequence of several molecules being together in being processed together, or do you need a pre-existing compartment where the SNRPs go there to undergo these specific modifications? And apparently, the structure is not necessary, because there have been several attempts of avoiding making knockouts of the protein called coiling, that is the scaffold of the Cajal body. And even without the body, the SNRP assembly did not undergo major problems. So it seems that the pre-existing body is not essential.
Very so we're going to hear you again after lunch. What? Okay. So thank you so much for really a lovely talk and for arranging this in such a short period of time. We are all missing you, giving you a big hug in Munich, and we hope to really see you soon again. We're going
Very good.
to be here again at 1 o'clock. No, 1 o'clock. 2 o'clock. Okay.
2 o'clock. 2 o'clock. Very good. Thank you. Thank you.
Bye. Carmo, do you hear me? Carmo,
Yes,
do you hear me?
I will leave the connection on so that we don't have more problems, okay?
Anything wrong on the computer? Where is it?
Close the camera and the microphone. Begin at 2.
Yeah,
Can you
but
put
I leave the connection on, okay?
Don't disconnect yourself. Remain as is, okay?
Sorry, should I?
With your computer connected.
Yeah, yeah.
Connected for 2 o'clock. Don't turn off.
Yeah.
Okay, thanks a lot, Carmo. I really miss not seeing you.
Yes, I'm so sorry. Yeah.
Yeah, never mind.
Yeah.
Okay, okay, see you, bye.
Okay, see you in 1 hour now.
א', יש לך פה תאורות, יש לך פה בשנה. אוקיי, אני בטוח שיש לך לייט.
כן. overhead
שם פה.
stage. No.
פשוט קח את הספוטים, לסובב אותם.
יש פה כפתורים, אנחנו יכול...
neon.
Okay, I hope you enjoyed the lunch, and I would like to remind you this quote from Sidney Brenner, who said, "Progress in science depends on new techniques, new discoveries, and new ideas, probably in that order." And the reason why I like a lot this statement is because I am truly fascinated by the potential of new technologies, how you can look for what was before invisible when new technologies emerge. And I would like to show you how this has influenced our ability to continue our quest about how fast is splicing, what is the timing of splicing in the cells. I showed you that we produce these live cell imaging measurements in that our results indicated a very fast splicing reaction occurring within a few seconds. But this was very controversial, because in the field, many other studies indicated that splicing would take a few minutes to occur. And many of the criticisms that I received, and these were all very fair criticisms, is that we were looking at a single reported gene. We can never be sure that the imaging procedures that we use are not interfering with the endogenous cellular process. So I thought, well, we should take another approach, use a different methodology to analyze the kinetics of co-transcriptional splicing. And we should, as much as possible, get an overview of as many genes as possible. So that's how I embarked on genome-wide studies. And for this, I was again very lucky to be able to start a very fruitful collaboration with Nick Proudfoot at Oxford University. In those days, Nick had a very talented postdoc called Taka Nojima. And Taka was adapting technology called nascent transcript sequencing, for short, NetSeq, that was originally pioneered, developed by Churchman and Weissman. And the basic idea is to isolate chromatin, to fragment the chromatin using MNase digestion, so that you get these nascent transcript complexes containing the polymerase attached to the template. So there is a bit of DNA in these complexes, and then the polymerase is associated with the nascent RNA that is being transcribed. So once we get these complexes solubilized, we can immunoprecipitate with antibodies against POL2. And then what we do is we isolate the RNA that is associated with the complex, and we do 3' end sequencing. So we sequence from the 3' and the 3' OH of each fragment associated with pole 2. As we started analyzing this data from HeLa cells using the NetSeq approach, we were very excited to see that in these complexes purified with antibodies against the polymerase, we could detect components of the spliceosome, we could detect splicing intermediates, so clearly telling us that splicing was intimately connected with the polymerase. And we also found a surprise because these antibodies, they can distinguish different isoforms of the polymerase depending on the modification status of the carboxy terminal domain of the larger subunit. So this carboxy terminal domain, or CTD, forms a flexible arm that allows for the binding of many proteins and is composed of repeats of these amino acids, and particularly the serine residues can be phosphorylated. But depending on the position of the serine residues that are phosphorylated, many labs have defined the so-called CTD phosphorylation code, which differs during transcription. So as the polymerase binds to the promoter, normally most of the CTD is unphosphorylated. And then as the polymerase initiates transcription, the phosphorylation occurs mostly on serine 5 residues. And then as the polymerase elongates, it's mostly serine 2 residues that are phosphorylated. So when we were looking at splicing associated with the polymerase, we were expecting to find mostly serine 2 CTD phosphorylation patterns because splicing occurs during elongation. But this was not the case. In fact, we found that most spliceosomes, even when we do mass spec analysis of the elongating complexes associated with polymerases with different phosphorylation patterns, there is a clear enrichment of spliceosome components, specifically with the serine 5 phosphorylation pattern. So clearly, the story of the CTD phosphorylation is not that simple, and as the polymerase elongates and splicing occurs, there must be shifts in the phosphorylation patterns that before were not clear. But we were a bit sad because we were finding very few spliced reads associated with the polymerase. And as we were doing these studies, the lab of Neugebauer published this beautiful Cell paper where they were looking at yeast and they were looking at co-transcriptional splicing, and they found that splicing was occurring immediately after the polymerase ended the transcription of an infant. So they could find polymerases transcribing the downstream exon, and splicing had already occurred. So why Carla and her group were observing spliced reads associated with these elongating polymerases, and we were not. Well, Carla was looking at yeast genes, and yeast genes have very short introns. In our NetSeq analysis, we have a limitation because we use MNAs to solubilize the complexes, and MNAs does not only fragments DNA, but also fragments the nascent RNA. So we are limited to short pieces of nascent RNA. And If in mammalian cells, because introns tend to be longer, one of the possibilities, one of the current models in the splicing field is that in order for exons downstream of very long introns to be recognized for splicing, the polymerase must transcribe the entire exon, and only then, when all the factors bound to the 3' in the 5' splice sites interact with each other, then the spliceosome assembles and splicing occurs. And maybe in our case, our short fragments due to mRNA's digestion were too short for us to see these polymerases downstream of the exon, and therefore we were not detecting splice streets. So in order to overcome this problem, we thought, well, maybe it would be informative to look at nascent transcripts in Drosophila embryos, because Drosophila embryos have this property that at very early time in embryo development, because the divisions, the cell cycle divisions are very fast, cells are dividing very fast, there is not much time to transcribe very long genes. So in the very early stages of Drosophila embryogenesis, most genes that are expressed are short, and they have short introns, and they are very reminiscent of yeast genes. But as the embryogenesis progresses and cell cycles become longer, then more human-like genes with longer introns start to be transcribed and spliced. So we thought, well, let's adapt the NetSeq protocol to Drosophila embryos, and these were the people. I had never worked with Drosophila in my lab, so we started a very fruitful collaboration with the Drosophila geneticist, Rui Martinho, and Pedro was a co-PhD student between Rui's lab and my lab, and Rosina is a very talented bioinformatician that did all the bioinformatic analysis. And so initially we could confirm what we had already observed in EULA cells, that when we use antibodies against the serine-5 phosphorylated polymerase, we do detect components of the spliceosome associated with the polymerase, and we detect splicing intermediates, namely the upstream exon that has already been cleaved, and the lariat that has already been excised. All of these intermediates, they have 3' OH, and therefore they can be detected by NetSeq. We could also detect recursive splicing in very long introns of Drosophila genes. Recursive splicing means that very long introns are spliced in pieces. So as the polymerase is transcribing an intron, a part of this intron is ligating to the upstream exon. So this portion of the intron is spliced out. And you can find these intermediates in NetSeq, giving us confident that we are indeed detecting the very nascent transcripts in the kinetics of what happens in terms of splicing. And now we were excited to see many more spliced reads in our Drosophila data sets. And we started by looking at short introns, and as expected, just like Carla had observed in yeast, we start to detect spliced reads as the polymerase is transcribing the downstream exons. So far, so good. But what about longer introns? So we move to those genes that are expressed later in embryogenesis in Drosophila, You know, in here you see a typical short exon flanked by two long introns. And again, we can detect splice reads as soon as the polymerase is transcribing the exon. So we observe over and over across the genome that immediate splicing, we call it immediate splicing to refer to this fact that splicing is observed right after the splice site is transcribed. And we do find immediate splicing across all intron sizes. There's no preference between, no difference between shorter or longer. And so this argues that maybe the exon definition model is not always required for splicing to occur. In this sense, I would like to remind you that when we think about the difficulty of joining two exons that are separated by a very long intron, we tend to think about messenger RNA as linear molecules. If a nascent RNA was linear, then in fact, this splice site would be very far away from the downstream splice site because the intron is very long. But if we think about nascent transcripts folded, and if there is any way that the upstream exon may remain somehow tethered to the polymerase, perhaps throughout the CTD, this is not clear, but if the upstream exon remains close in space to the polymerase, then it doesn't matter to have a longer intron because the intron will fold around and will not interfere with the proximity between the splice sites that are spatially positioned to be ligated irrespective of the intron length. And in fact, this idea of exon tethering was proposed by Sue Beck in the year 2000. So as I told you, we are always eager to progress with our techniques and eager to overcome the limitations of our techniques. And I told you that digesting the nascent transcripts with mRNAs is introducing a limit for size. So more recently, Taka and a PhD from my lab, Rui, have been trying to improve the NetSeq technology, getting rid of the mRNA's digestion component. And Taka came up with a new method that he calls point for Pol2 associated intact nascent transcripts. And now the Pol2 and their nascent associated nascent RNA are removed from, are solubilized from chromatin by DNase digestion. So the nascent RNA molecule remains intact. And now this is again soluble and can be immunoprecipitated with antibodies against the Pol2. And now that we have intact RNA molecules, we can make use of the next generation sequencing technologies, which are single molecule long read techniques. Nanopore is one of them, PacBio is another. But the big advantage is that you can now sequence the entire length of the RNA instead of the traditional Illumina sequencing where you have to fragment and then you sequence the fragments of the RNA. And when we look with these long reads to what happens in terms of splicing along a gene, this is just one example, we do see reads. Look at these red reads. These red reads correspond to mRNA molecules from the very beginning, from the TSS up to the middle of this exon. So the polymerase is transcribing the exon and the RNA has already been spliced. So these reads are spliced. All the red reads are spliced, confirming that in mammalian cells, yes, splicing can be immediate. You don't need to transcribe the exon in order to assemble the spliceosome. But what was very surprising was that we do see spliced reads, but look at all these blue reads. The blue reads are unspliced, and they are many more than the red reads. In fact, the majority of pre-messenger RNA it remains unspliced, and this is not intron retention. This is not retention of a particular intron. Is that the entire mRNA, the entire pre-messenger RNA, all introns remain unspliced. So this is quite surprising. So does this mean that a lot of the transcripts are just wasted because they miss splicing? And also that there seems to be here something counterintuitive, because on one hand, we see splicing as an extremely efficient process that can occur as the polymerase transcribes past a splice site. And yet, splicing seems also to be a very inefficient process because many of the transcripts just miss the splicing. How is this coordinated? Is there any function? Are these pre-messenger RNA just wasting? Is this a stochastic decision to splice or not to splice? So these are all remaining and open questions that we came up with. And I would like to reinforce this view and this fascination of mine of looking at the same biological problem with new techniques is always uncovering surprises. And with this, I leave you with my current group, and I'm looking forward to your questions.
I have a question. You hear me?
Yes, I hear you.
And I was surprised when you said that the transcript is spliced after it's transcribed the 3' splice site. Because if this would be if this occur in every case, how would you consider alternative splicing?
Yes, yes, this is.
Splicing, you cannot do that.
Yeah, absolutely, absolutely. But in alternative splicing, it's the decision whether a particular exon is skipped, then obviously the decision to skip an exon must be taken well before the polymerase starts transcribing that exon. Otherwise, it would never be skipped.
That's one possibility. Another possibility is that those cases that are not spliced are having time to, in case you need alternative splicing, to choose the right junctions. Did you try a new method with transcripts, specific transcripts that undergo alternative splicing, and
Yes.
those that not go alternative splicing?
Absolutely, you are just touching on the sensitive point. So those transcripts that we see unspliced, it's not restricted to alternative splicing. It's all the introns. It's like a all or none. Either we have splicing, efficient splicing, very fast splicing, or we don't have splicing of the multiple introns. Now, we do not yet have good coverage of alternative splicing events. And this is the next frontier, because a limitation of these long-read sequencing methods is that they have very low coverage. So we are still struggling to get more data on all types of isoforms. In fact, this is now the big trend in the splicing field. Lots of labs are optimizing these long-read sequencing methods to be able to make maps of all the isoforms, of all the alternatives evenly spliced isoforms that are produced in each cell type. So combining long reads with single cell transcriptomics to see what isoforms are being made in each cell type. This is on one hand. For us, we are still struggling to get enough coverage so that we can tell what's going on with alternative splicing events. But you are absolutely right, Ruth. This does not explain everything.
Are there any questions, other questions? If not, thank you so much, and I really miss you. Third one, there's one, yes, please. Quick one.
Can you hear me?
Can you hear?
Yes, I can hear you.
So I don't know if there's a way to analyze transcriptional coding in high resolutions. But has anyone looked if longer introns are followed by code sites that would allow the formation structure that would join the far end of the intron?
Your question is about pausing of the polymerase.
Right.
Yeah. So I didn't mention anything related to pausing of the polymerase because this is currently another controversy in the field. Because again, using different methodologies, using NetSeq, we do see pausing of the polymerase associated with immediate splicing, just like you are asking. Probably the polymerase slows down in order to allow some time for the splicing to occur. But this we observe using NetSeq. But other labs using GROSSeq, they don't see any evidence for polymerase pausing. So I didn't want to go into this controversy, but this is a hot question in the field currently.
Thank you so much, Carlo. We really miss you. And after this talk, definitely we could have talked and had many discussions, fruitful discussions, but we have to move on and really appreciate.
I wish you a great course, and thank you, Ruth, for organizing all these.
Thank you. Our next speaker is Lorenzo Charles from the Maryland University. And Carmo has already mentioned his seminal work.